Dear students, good morning to you all. We are seeing that is human leukocyte antigen and mixed lymphocyte reaction. Human leukocyte antigen popularly is called human HLA. HLA. That is human leukocyte antigen. See, leukocyte. Leukocyte means we are having neutrophils, basophils, eosinophils. Okay, so macrophages, dendritic cells. All these different 5 types of the cells are having this antigen. Apart from that, there is HLA1, HLA2 is there. Those details we see. And MSC is mostly being represented in mouse. In humans, it is called HLA. Both function is same. Okay. So the other one is mixed lymphocyte reaction. Mixed lymphocyte reactions. Whenever I want to donate my kidney to the other person. Okay, my blood cells and his blood cells are being taken. My WBCs and his WBCs. Both the things are cultured together. That's what mixed lymphocyte reaction. In this culture, if everything goes well, they will not proliferate. If these two cells find that they are different, then each cell proliferate, and once they proliferate too many cells, then they are not compatible. They are not compatible, we have to do something. That's what mixed lymphocyte reaction is being done before the transplantation of organs. Okay, so the human leukocyte antigen, HLA, is having a lot of prominence. HLA is said to be the self, that is, and our own antigen. Okay, so what is happening is, in immunology we used to say, what is immunology? We say, tolerate self, target non-self. Tolerate self, target non-self. What is self? Self is HLA molecule. Because that is your HLA, my HLA is different. So your HLA becomes the target for me. My HLA becomes target for you. Okay, so that's what now my HLA molecules and your HLA molecules are not compatible. Okay, all over the world, that's what the situation. Otherwise, the organ transplantation can be done by taking from one individual, it can be given to the other individual. Like that, there will be big massacre. But it's not so. Nature doesn't permit to transplant organs as we like because of the HLA. Okay, human leukocyte antigen. So, human leukocyte antigen is being also studied with regard to the human migration. In that also it is having some importance, and a group of individuals, ethnic group of individuals are having one set of HLA molecules. Even if you plan for transplantation, we have to plan only from our ethnic group. Power of group may not be compatible. So like that, there is plenty of things are there in relation to the human leukocyte antigen. Hitherto I have been telling to you about the transplantation, but in addition to that, it is having another major role in elicitation of the immune response. Elicitation of the immune response. That is, whenever if a coronavirus is attacked, coronavirus is what is it called? Digested by the dendritic cell. This dendritic cell, as I told you just now, leukocytes are having this HLA molecule. They present this coronavirus antigen to T cytotoxic lymphocyte through MSC molecule, through HLA molecule. That is, ultimately what I wish to let you know is HLA molecule will be useful for presenting antigen to T helper cells, T cytotoxic cells. This is another important point. So that's what whenever the vaccine is being designed, we also have to take into consideration the vaccine peptides. Do they get anchored to HLA molecule or not? Okay, so several applications are there, human leukocyte antigen. Let us see the details now.
Now let's talk about human leukocytic antigen, or HLA. They are called antigens, so they are antigens. Where do you find them? On the surface of the white blood cells. Where? In humans. In other words, they are human version of the major histocompatibility complex. Why did we call it major? Because there is minor histocompatibility complex. Why complex? Because it's more than one protein. Why histocompatibility? Because it's compatible with tissue. Translation: the human version is different from the monkey version, for example. You're saying that the human leukocytic antigen in human is different from cockroaches? Yes, honey, that's precisely what I'm saying, and this is the whole freaking point. That's why when someone goes to undergo an organ transplant, they do the MHC matching before. Is this new tissue compatible with your tissue? We have 3 classes of MHC, class 1, class 2, and class 3, creative. Okay, class 1, they include HLA, A, B, and C. They interact with the T lymphocyte. Please be specific. The cytotoxic CD8 positive T cell. How about class 2? They have the HLA-D, and we have DP, DQ, DR. If you remember your AKG, the first wave was the P wave. We have the P, and then we have the Q, R, S complex, and then whatever. So DP, DQ, DR, and then the DR has subtypes DR2, DR3, and DR4. These interact with the T lymphocytes. Please be specific, the helper CD4 T cells. If you pay attention, these interact with T lymphocytes and these interact with T lymphocyte. So you're saying that it's only cell-mediated immunity and not humoral immunity? Yes, sunshine, that's exactly what I'm saying. You know, passive immunity is actually garbage, just randomly fighting stuff. Yeah, it's better than nothing, but it's still trash, like your natural killer cells, haphazardly beating the crap out of everything without accuracy, let alone precision. Then, at a higher tier, comes your B lymphocytes, a higher, newer generation. They secrete antibody in the fluids of your body. Fluids, that's why we call it humoral immunity. Humoral in medicine means fluid. That's why B lymphocytes action are called humoral immunity. Better than natural killer cells, still not that great. Then at the highest tier we have the T lymphocytes, a great new level of specialization, the apex of specialization as Adam Smith would describe it. Comes the T lymphocyte, a new world of accuracy and precision. We call it cell-mediated immunity, and that's what we're talking about when we talk about the HLA. Only T lymphocyte, no B lymphocyte. Some mnemonics. Class 1, they interact with CD8. Class 2, interact with CD4, the T helper cell. 8 times 1 is 8. 4 times 2 is 8. The result is always 8. And this is arguably the oldest medical mnemonic known to man. When God created Adam and after he named the animals, he invented the mnemonic for the major histocompatibility complex and passed it down to future generation together with the X chromosome. I think I'm a stupid idiot. I'm recording under influence. Okay, let's do it from scratch. We have 1, 2, 3. In this direction we have the MHC, and in this direction we have the HLA. So 1, 2, 3, A, B, C. Here we have D. Also 3, P, Q, R. Start with the AKG, which has 3 letters. P, Q, R, and then R has 3 subtypes, 2, 3, 4. Let's do it again. We have MHC, 1, 2, 3. We have HLA, A, B, C, D. DP, DQ, DR. The DR has 3 subtypes, DR2, DR3, DR4. Unfortunately, there is DR5 and other stuff, but let's use the KISS principle and keep it simple, stupid. Let's make it more complicated. Human leukocytic antigen, the human version of the MHC. Class 1, class 2, we don't care about class 3 right now. We have A and we have B. How about HLA-A3? It's associated with hemochromatosis. Remember, the iron has the ferric, and the ferric is 3+. Remember, A3. Just the mnemonic, guys. Come on. HLA-B, we have HLA-B27, associated with the seronegative spondyloarthropathies, which we'll talk about later. And HLA-B8, Graves' disease, Addison's disease, and myasthenia. The cruel autoantibodies ate my thyroid, my adrenals, and my energy.
Okay. Now we have gone through a video showing HLA, wherein HLA class 1, class 2, class 1 is having ABC, all those details are there. Those things we are going to see now also. So the objectives of this particular topic on HLA and MLR, human leukocyte antigen and mixed lymphocyte reaction, is as follows. To understand the types of HLA molecules, to acquaint with the nomenclature of HLA, to be aware of HLA genes and their location, to appraise HLA polymorphism in the population, and to appreciate the role of HLA in transplantation through MLR test. Role of HLA in transplantation through MLR test. Okay, to familiarize the role of HLA in antigen presentation. So these are all the various things that we are going to see now in this particular topic. So to see the nomenclature, HLA nom, because it is so many are there. That is HLA class 1, HLA class 2. Okay, and again is HLA class 1, there is HLA class 1 A, B, C, E, F, G. There is no D. A, B, C, E, F, G. And A is not only one. A is having again more than 200 or 300, what is it called, the subtype. And B is having, so B is highly polymorphic, more than 1000. So like that, various numbers are there, alleles, various alleles in this HLA molecule. And again, HLA class 2 is having DP, DQ, DR, DF. Okay, and again in this DR is highly polymorphic. There is several alleles are there in DR. So that's what there is a fixed nomenclature here. Okay. So HLA genes, there's a set of HLA genes which are present on chromosome 6. Those details we see now. Polymorphism, just I was mentioning to you. MLR test. So this is antigen presentation. So HLA polymorphism is so important that nature has made us in such a way that if we are because there is polymorphism, organ transplantation is not possible. Okay, so it is possible only when there is matching. Matching is very rare. So expected outcome, able to distinguish between self and non-self molecules, type, structure, genes, polymorphism and distribution of HLA class 1, class 2 will be known as a prelude to the surgical organ transplantation. The role of MLR test in organ transplantation will be known. So this is one of the crux and important aspect of immunology. What is HLA? What is it for? It is a human leukocyte antigen. Leukocyte means we all know they are blood cells. That is, in the blood cells are classified as leukocytes and lymphocytes, and these leukocytes are having specific antigen called HLA antigen. They are said to be self molecules. So major function of this in our immune system, what is the function of HLA means? To present foreign peptide to T cell. That is the basic function. As a result, the immune response is excited, thus allowing aroused immune response and elimination of the foreign particles. So that is the main function of HLA, to present foreign peptide to T cells, thus allowing aroused immune response and elimination of foreign particles. Okay, so now let us see antigen presenting cell, that is dendritic cell. It is having MSC molecule. Through the MSC molecule, it is holding a peptide, and that peptide, along with the peptide, MSC molecule is presented to T cell. T cell is having T cell receptor complex called TCR. So that is the major function of MSC. Okay, without MSC, antigen is not being seen by T cell. Okay, now there is T cell. T cell can see the antigen. You can have a question: when are the T cells see the antigen which is freely floating in our system? No, T cell cannot see the antigen. T cell can see the antigen only it is presented through MSC class 1 or class 2. Okay, so as it is simply, if anybody wants to serve the food, they serve the food in the plate. If there is no plate, we can't take the food. That is the purpose here. Okay. So presentation, peptide to T cell. So MSC class and HLAs are glycoproteins. They are the glycoproteins. Because glycoprotein, if it is any of the mannose and some of those things get attached to the protein, means protein is stable for a long time. Okay, central molecule. These are the central molecules in immune information. The immune system, they convey immune information. This is HLA glycoprotein 1, HLA glycoprotein 2. Okay, so HLA glycoprotein 1 is expressed in all nucleated cells, brain cells, kidney cells. That's what, whenever there is matching is done, kidney matching is done, HLA class 1, HLA class 1B, particularly B is matched well, because B is highly polymorphic. Okay, and HLA GP2, that is glycoprotein 2, is expressed on antigen presenting cells. They are nothing but leukocytes. Okay, so basic function of HLA is information conveyance, that is peptide conveyance, antigen peptide conveyance. So HLA class 1 is present in all nucleated. That means RBC doesn't have. RBC does not have the nucleus, so HLA class 1, HLA class 2 are not there on RBC. Okay, HLA class 2. Okay, let us see the further divisions as I was mentioning to you. HLA class 1 is having A, B, C, no D there, E, F, G. B is highly polymorphic. Okay, and G, see, please get to know the G. See, the fetus that is growing in the womb of the mother is tolerating that fetus. Though the fetus is having half of what is it called, the father's chromosomes. Their father HLA is different, but still the mother is holding the fetus. Why? Because HLA in the fetus only G is expressed. A, B, C, E, F, G, F are not expressed in the fetus. G is expressed. That G is least polymorphic. That is almost homozygous, least polymorphic. That's what the mother is accepting, that fetus having the G being expressed there. Later, once it becomes infant, it expresses, it will not be expressed. That's what in most of the tissues, there's differential expression of genes. In the fetus, it's expressing. That's what in the fetus also, with regard to the hemoglobin, if you take hemoglobin Ig, there is a hemoglobin Y is there. Once we come to the adult, we are having hemoglobin, alpha hemoglobin and beta hemoglobin. But in the case of infant, it is gamma hemoglobin. Okay, so that's what differential expression of genes are there in the developmental stages. That's what HLA-G is being expressed, which is least in polymorphic, that is being accepted by the mother. Okay, so that's what here HLA-GP2 expressed on all antigen presenting cells. DP, DQ, DR, DM. DR is highly polymorphic. Just for the reference of telling you, I made it in a different color. Now let us see, these are all the various distributions. And HLA class 1 is present, and all the cells see, all are plus marked here. HLA MSC class 2 is present, basically in antigen APC, antigen presenting cells, and thymus epithelium. In the thymus epithelium, because they are required for the thymic schooling and education. Okay. Now, this is human chromosomes, a character arranged just for the purpose of showing this HLA genes complex is present on chromosome 6. We have got 23 chromosomes, of which 6 chromosomes it contains. Now, let us see the expansion of this chromosome. So, that is the centromere that divides into P arm and Q arm. You have studied in classical genetics. So, in the P arm, where the red mark is given, the HLA complex is present. Okay, the HLA complex is present BCA. And here DP, DQ, DR, like that they are arranged in the HLA complex and the P arm of chromosome 6. Okay. Now let us see some more details. So this is the HLA class 2 region, DP, DQ, DR, DM. And again alpha chain, beta chain, alpha chain and beta chain. So here this is the HLA class 1 region. HLA B, C is there. And down if you see HLA A is there, G is there, HLA F is there. So like that they are distributed across the gene length in the chromosome 6 in the P arm. Okay. Now the structure of HLA. So they are the glycoproteins, they are heterodimers. Heterodimers means it is not a single protein, it's having alpha chain, it's having beta chain, it is having beta microglobulin in some cases. So like this is heterodimer. It is heterodimer, like that hemoglobin is having 4 polypeptide chains. Okay, 2 alpha and 2 beta. 2 alpha chains, 2 beta chains are present in the hemoglobin. Like this, HLA is having alpha chain and beta chain. But HLA1 is having beta 2 microglobulin. So that's what is the heterodimers. Okay, structure of HLA molecule of both classes enables antigen binding. So particularly we are having HLA so that antigen gets anchored to HLA. Antigen gets anchored to HLA. Okay, so this HLA with the antigen is being presented to the T cell receptors. That's the main purpose. This HLA is made in endoplasmic reticulum. Later, it is placed in the extracellular. Okay, it is highly polymorphic. Predominantly, the cleft region is polymorphic. Cleft of the HLA molecule. We are showing the diagram now. Highly polymorphic, particularly in the cleft region. Okay, non-polymorphic portion of the molecule is there in the binding sites. That's related to the CD4, CD8, not in the cleft. So now we are seeing the HLA molecule. It is a pictorial representation. So wherein we are seeing alpha 1, alpha 2, alpha 3, beta 2 microglobulin. That's what is called heterodimer. That is with regard to HLA class 1. Next one, if you come, it is alpha 1, alpha 2, beta 1, beta 2. That's 4 molecules are there, protein molecules. It's what heterodimer. Now if you take up HLA class 1, there is a cleft on the top that is shown by the arrow. The cleft accommodates the antigen. The black one is an antigen, just pictorial representation. That is the region. So that antigen is being presented to now the T cell. Where from that antigen has come? That the dendritic cell has given that antigen. Okay, we'll see that. This is the crystal structure of peptide binding to the cleft. That is the peptide red one. This is the crystal structure, foreign peptide, that is kept in the peptide. In this, what is it called? HLA cleft, in the HLA cleft. Okay, so the HLA cleft is having the, what is it called? The proteins made up of the anchoring residues, amino acids. Those amino acids bound, anchor to the peptide, foreign peptide, like that presents. Okay, so this is the crystal structure. So if you are, this is another way of showing, that is all the ball structures of the protein shown in PDB, protein data bank, where the alpha chain and wherein peptide is being attached. So the lower portion, if you see, this is a MSC class 1 having this, it accommodates only small peptide, green one you see. In the second diagram, if you go through MSC class 2, it accommodates a bigger green dimension, that is bacterial, that is peptides it accommodates. It is more than 13 amino acids. Here, these peptides are only between 8 to 10 amino acids. Okay, so that's what the arrangement between class 1 and class 2 MSC molecules. Class 1 MSC molecule is primarily for viral antigens, viral peptides. Viral peptides are small. Class 2 molecules are primarily for bacterial peptides, which are a little bit big, 13 amino acids or more. Okay, so that's what I wish to show you here. The size of the peptide that the MSC class 1 accommodates, the size of the peptide that MSC class 2 accommodates. Okay. So this is the nomenclature. As I was mentioning to you, HLA-B is highly polymorphic. Okay, in HLA-B itself, we have got more than 500, 600 alleles. Okay, let us take HLA-B 8 allele. Okay, 8 alleles have again sub divisions like HLA-801, HLA-802, HLA-821. Like this, it is enormous, bewildering diversity. That's what your HLA, my HLA doesn't match. That's what HLA is said to be self molecules. Like that, if we take a HLA B27 down, B27. So it is having B27, 01, 02, 03, and HLA 27, 25. It is a HLA B27 having the 25th subunit. So like that, there is enormous variation, so that one fellow HLA does not match the other fellow's HLA. Okay, so this is a little bit complex to get to know. So now, we are all human beings, living separately and all that, we are all maintaining this type of the diversity. But when you go to the wild animals, particularly cheetahs, tigers, they are living in, what is it called, museums and all, zoo parks and everything, where what is happening there, there is, what is it called, their homozygosity is maintained. Homozygosity is being maintained. That is, they are all internal breedings. Okay, they are breeding with their own family groups. So for generations together, they are breeding with their own family groups for their generations together. As a result, they maintain the HLA almost has become almost similar. There's no polymorphic. There no polymorphism. No polymorphism is prevailing. If there is no polymorphism, it is dangerous. They cannot survive. Species cannot survive. That is, in the population, only heterozygosity. In the evolution, the natural selection, heterozygosity is given the preference for survival species, for the survival of the species. If it is homozygosity, there is danger of survival, because homozygous individuals, if one individual gets infected, all of them get infected together, because they are all homozygous towards the HLA. So that there is a possibility for the collapse of the species. So that is one of the reasons for these wild animals gradually becoming extinct. Okay, so like that also we can study. So now we see I collected this information from the literature just for the purpose of you to know HLA is being spoken with the ethnic population, our Andhra people, our Guntur people, like this. So in Andhra Pradesh, HLA population, that is the population containing the HLA haplotypes are like this: HLA A1, B57, DR B17. So that is the combination I am having. So the other person who is there in Guntur may be having HLA-A33, B44, DRB107. So that is the combination. So this combination goes. That's what called haplotype as it is. There is no question of linkage and crossing over taking place. So that HLA-A33 cleaving from B44 doesn't happen. HLA-A33 goes with B44. And HLA-A33B and B44 goes with DRB107. That's what haplotype, one cassette. One cassette of the DNA, it doesn't undergo crossing over, it doesn't undergo recombination, it gets inherited as it is to the offspring. That's what called HLA haplotype. Okay. So let us see the HLA polymorphism. Now as we have seen, HLA-B, MSc class 1, look here, wherein B is highly polymorphic, having 300, more than 300 alleles. C is least, A is having 195, and here HLA-2, there is DR. DR is highly polymorphic. Still these numbers are high now. Okay, so that's what. So many alleles, 323 means so many alleles are spread across. What allele I am having, you may not be having. Okay, so that's what it is highly bewildering. So with 4 nucleotides, adenine, guanine, cytosine, thymine, we have got millions of genes. With 323 combinations of what is it called DR, and 395 combination alleles of B, HLA-B, and again 195 combination of HLA-A. How many combinations do we come across? It is not imaginable. So many. Okay, so that's what called the HLA polymorphism. Okay, so during the transplantation, if my HLA matches to you, then it goes well. If it doesn't match, there is recruitment of cells and there is graft rejection takes place as it is shown in the second one through the arrow. Okay. So HLA matching between the donor and the recipient, a predictor of a graft survival in the kidney allograft patient. So whenever we speak the transplantation, whenever we say, do you have the immune system, means yes, I am having the immune system, I reject allograft. You have to say, I reject allograft. Allograft means the graft that is taken from the other individual. Okay, that other individual is different. He is not your cousin. He is not your, what is it called? Twin brothers. He is not your twin brothers. Okay, so the brothers who are born from single zygote. The brothers who are born from single zygote. The zygote at the time of the development, they cleaves. As a result, each portion of the zygote cleaved into two, they become twins. They are isozygotic twins. They are similar, ditto in all aspects. So that is not called allograft, that's called isograft. Okay, so this is allograft means this is another individual who is having a different HLA molecule. So that type of allograft I reject. That's the reason why I am having the immune system. Okay, HLA mismatches may cause chronic pathologies in the graft. It is clear that a negative HLA-A2, if I am having negative towards HLA-A2 recipient, receiving an allograft expressing HLA-A2, that is, if I receive an allograft, he is positive to HLA-A2, then much higher risk of humoral rejection of the vascular endothelium. So for which the doctors for the time being they are giving the cyclosporine, such type of immunosuppressive drugs, and again they are also giving anti-CD3 treatment, that is antibody against CD3, because T cell is the one which maintains all this what is that called regulations, because the master immune cell. So they make master immune cell to get suppressed. So some of the treatment they give if it gets adjusted. Okay. So now let us see the mixed lymphocyte reaction, MLR. So this mixed lymphocyte reaction we are doing to confirm. Mixed lymphocyte reaction we are doing to confirm HLA is matching or not. HLA is matching, nearly matching or not. Okay. So determination of T lymphocyte alloreactivity. Mixed donor recipient lymphocyte T lymphocytes after recognition of allogenic MSC glycoprotein activate and proliferate. Okay. So one way MLR is there, two-way MLR is there. Mixed lymphocyte reaction. One way MLR, two-way MLR is there. Okay, so in one-way MLR, a recipient cell is made. What is it called? It is made a sort of non-mitogenic. It is made as non-mitogenic. It will not proliferate. So it is irradiated. Recipient cells are irradiated. Okay, so when these recipient cells are presented, the recipient, the donor cell, and all these things reaction takes place that we'll see now. Okay, again I will explain this mixed lymphocyte reaction. Okay, one way MLR. One way MLR means the donor cells are irradiated so that they will not proliferate. Recipient cells are kept as it is. So when the donor cell and the recipient cell are kept together, because donor cells are unable to proliferate, recipient cells are fine. If they identify the donor cells are nearer to them or same to them, they will not proliferate. If it is far away with regard to the HLA molecules, the recipient cells proliferate and they try to kill the donor cells. So that's what one-way MLR. Two-way MLR means both are not, what is it called, irradiated. So there the differences we may not be knowing. That's what doctors prefer, one-way MLR. Okay, T-cell reactivity. So please see here, this is allele A donor, A fellow. It donates. The cells are irradiated. That's what I was telling to you. Okay, so here we match to the allele A recipient. So they're all going well. There's no reaction. There is no reaction. If it is given to the allele B cells, there is activation and proliferation of the recipient cells. That is the B cells. See the diagram. B cells are proliferated. As a result, and that B cell proliferation, just like that in the microscope, we may not completely see. We put tritiated thymidine. So if we estimate the tritiated thymidine through this radioactive counters like GM counter, scintillation counter, we'll be knowing the value. So that is the way we'll do the mixed lymphocyte reaction. Okay, so like that we'll assess patient A and a donor. Patient A donor are matching or not. Okay.
Hello everyone and welcome to today's video. In today's video we're going to be discussing the mixed lymphocyte reaction. So it is a technique used to assess whether an individual is going to be undergoing an immune response against another individual's cells or graft. So, I mean, naturally, we know from all the other episodes that in this series, we know that, generally speaking, unless it was an autograft or an isograft, the immune system is going to be eliciting a reaction. However, it is important to know to what extent this reaction is going to be, or this rejection is going to be. So, the steps are as follows. First of all, we're going to be taking a sample of mononuclear cells from two donors, the X and the Y, the ones that we want to study whether there's going to be an immune reaction or how severe the immune reaction is going to be. Now, we're going to be making sure to add the mitomycin B or any other chemical that is going to be mitotically activate the mononuclear cells of Y. Now, this is why it's important. Essentially, we're going to be having a very limited number of Y cells, and these Y cells are going to be the ones that the X cells are going to be binding to and eliciting a reaction. We'll understand that as we progress in these steps. Next, we're going to be mixing them up. So now we have a lot of, well, to an extent, since we are, these are going to be clonally expanding after they recognize themselves. Hello everyone and welcome to today's video. In today's video we're going to be discussing the mixed lymphocyte reaction. So it is a technique used to assess whether an individual is going to be undergoing an immune response against another individual's cells or graft. Now this is not very accurate because generally speaking we know that autografts and isografts are going to be they're not going to be inducing any immune reaction, and the others, be it xenografts or allografts, they are going to be inducing certain reactions. However, it is very important to know to what extent these immune reactions are going to be. So the steps are as follows. We take a sample of the mononuclear cells from two donors, the X and the Y, and we make sure that one of these two are going to be, we're going to be adding some some chemicals that are going to be preventing the mitosis of these cells. Now, next we're going to be mixing them together. The donor's X CD8, that means the cytotoxic T cells, are going to be binding. As we all know, the CD8 plus are going to be, or the cytotoxic T cells, are going to be binding to the MHC class 1 of the donor Y, APCs, while the donor's X CD4 plus, that means the T helper cells, are going to be binding to the MHC class 2 also naturally. Now, what is going to be happening here is that after clonal expansion, so now they know that there is a problem. They know that these are not self, right? Because they bound to them. Since we can deduce that when they when they bind to these MHC class 1 and the MHC class 2, we're going to be inducing different types of reactions. And this is what we're going to be discussing now. So after clonal expansion, I mean, obviously, we're going to be having clonal expansion because these cells, they're not going to be, how can I say this? Only a few cells are not going to be taking care of the whole body. So naturally, our cells are going to be expanding in number, and they're going to be specific against this particular cell. So after clonal expansion and maturation of the CD8 plus, that means the cytotoxic T cells, they're going to be binding to the MHC class 1, and as we all know, cytotoxic T cells are going to be killing these cells. And CD4 plus are going to be binding to the MHC class 2, and what they're going to do, well, we know that CD4 plus or the T helper cells are going to be releasing cytokines. These cytokines are going to be leading to many problems that are going to be discussed in a future video. These actions are going to be these actions will indicate that if we were to graft cells from a donor to a recipient, they would get rejected. Now, this concludes this today's episode of Transplantational Immunology. This has been Ali from B-Biology.
So now we see the video on MLR just for more information purpose. Okay, now we have gone through this MLR. It's quite interesting. So with this we go to the summary. Function of HLA while presenting a foreign peptide to a T cell is elucidated. Types of HLAs are made familiarized. And location of HLA gene complex on chromosome 6 between 6p21.1 to 6p22.1 cm centimorgan is shown. Structure of HLA including cleft geometry is displayed. We have seen HLA class 1, class 2 molecules, and HLA haplotypes of Andhra Pradesh and its nomenclature are appreciated. The role of HLA in organ transplantation is shown. So study questions: what are HLA? Explain their structure and function. How do HLA 1 and 2 convey information to T cells? This is what for arousal of the immune response. Enlist distribution of HLA 1 and 2 in human cells. Indicate the gene location of HLA gene complex. Where does the HLA gene complex locate in the chromosome? Elaborate on HLA polymorphism and HLA haplotypes in the state of Andhra Pradesh. Okay, so let us sincerely acknowledge online resources and public domains from which we use the contents to develop teaching material. Thank you, Ananda. Please go through this topic. Thank you.